So thank you very much, Ralph, for the introduction. I'd like to start by thanking the organizers of this workshop and the organizers of the program in general for inviting me here and putting this on. It's been very enjoyable. I'm also going to start with a bit of an apology. I don't know if anyone's read my abstract, but it's sort of as non-committal as I could make it, and that's because I didn't know what I was going to talk about until a few days ago. And the work I'm actually going to talk to you about is work that we started while we were here in the Newton Institute in March. So it's still work in progress, but I think it's relevant to this program, and it's joint work with Charlie Elliott and Tom Renner. So we're going to be interested in receptor-ligand dynamics, and the idea here is that you have receptors which live in an extracellular medium around the cell, and they're going to want to react with ligands that live on the cell surface. So you should just think of this green cross-section here. Ignore everything that's inside the cell. And what happens is when these receptors react with these ligands, they form receptor-ligand complexes. And so there's an on rate associated with this reaction, which is this rate K2, and there's an off rate, which measures the rate at which these receptor-ligand complexes dissociate to release ligands back to the bulk and release a free receptor back on the surface. And so we can come up with a very simple mathematical model for this process. And so I have my bulk domain, omega, which you should think of as being the extracellular matrix. And I'm going to assume there's a uniform background source of ligands on the outer boundary of omega. Okay, so that's effectively a Dirichlet boundary condition on the outer boundary, on this boundary d0 omega. And I'm going to assume that the ligand concentration satisfies the heat equation inside omega. And then I'll model these receptor-ligand dynamics by assuming that there's a flux, the normal derivative of the ligand concentration is given by these two terms here on this inner boundary gamma, which is meant to denote the cell membrane. So you have loss of bulk ligands as they form these receptor-ligand complexes, and you pick up bulk ligands as the receptor-ligand complexes degrade to release a ligand back into the bulk. And then coupled to this PD in omega, we have two PDs on gamma, one for the concentration of surface receptors, which are assumed to be mobile on gamma, at least at the start of this talk. And again, you'll lose receptors when they form receptor-ligand complexes, and you'll gain them when the receptor-ligand complexes break down. And you have a similar equation for the receptor-ligand complexes. This is a toy model. I mean, in practice, in biology, things are much more complicated, and we're not going to try and deal with models like this, but actually, a slightly more complicated model, which is still an oversimplification, is to say that receptors and ligands can also be internalized, and this is an Elie-Riedel mechanism for the whole process. So basically now you have extra PDs that are posed in the interior of the cell, and there you have PDs for the concentration of internalized receptors and internalized receptor ligands, and you get extra contributions to your surface PDs. Even this is an oversimplification, because what actually happens in practice is that the surface reaction is not occurring in the way I've explained to you so far, but what's happening is that ligands, when they approach the cell, they're more likely to encounter what's called a free binding site rather than a receptor. So these ligands first find a free binding site, and then they undergo what's called a conformal change, and it's irreversible, to become a surface ligand. And then finally, the surface ligand is transported by lateral diffusion over gamma to find a receptor to form a receptor-ligand complex. The whole model, I'm not going to write it down, but it's sort of 8 coupled PDs. You have 1 in omega, 5 on gamma, and 2 in the interior. But we're not going to see that model at all, so don't worry about it. There are lots of applications of these types of models. So one is chemotaxis. So in chemotaxis, what happens is that cells migrate up a chemical gradient, and one hypothesis for the way that they do this is that they sense the level of receptor occupancy across the cell membrane, and they move in the direction of highest receptor occupancy. So this movie here is dictyostelium cells all migrating towards a source of cyclic AMP in the middle, and it should be going a bit faster than this. I can make it go faster. Eventually, you see, you get something which looks potentially like, I guess, blow up in the Keller-Siegel model or something like that. We can also play at trying to simulate these equations. We weren't quite as ambitious with the number of cells. We had about 10, but they're all going to chase the source of chemoattractant in the center, and they adhere with each other while they're doing so. You could also look at things like substrate adhesion. Again, here, receptor-ligand dynamics play a really important role, because what happens is that the cell, it forms what are called focal adhesions with the substrate, and the way it does so is by binder proteins on the cell membrane bind to ligands on the substrate. If you disable this ability of the cell to form these focal adhesions, they can't migrate successfully anymore. So it's a crucial component of actin-based cell motility. And again, we've had a look at very much toy models for simulating these types of behaviors, where we have a PD on gamma for the surface binder density. There are lots and lots of other applications. I mean, one which I guess we heard a bit about in the talk earlier today is binding affinity in drug development. So one of the measures of the efficiency of a drug is its binding affinity with its target receptors on cells. And so there are lots of other areas in which understanding receptor-ligand dynamics is important. But for the purposes of this talk, we're not going to look at a biologically realistic model at all. We're going to focus on this very simple model that I introduced on the first slide. And I'm going to be interested in the case where this on rate, this parameter K1, is very large, much larger than the off rate K2. In fact, much larger than all the other parameters in the model. And so an outline of the talk is that I'll introduce the problem one gets when you consider this fast reaction rate, and we'll prove existence of a solution to this problem. And then I'll look at the problem one gets when you take the reaction rate to infinity, and we'll encounter what I'm going to call the parabolic limiting problem, and we'll analyze that. Then formally we'll drop certain terms, which will give us an elliptic limiting problem with dynamic boundary conditions, and we'll prove existence and uniqueness of a solution to that. And then finally we'll show that the elliptic limiting problem can be reformulated as a variational inequality where time enters as a parameter. So the fast reaction problem, I'm going to make my life even simpler, and I'm going to completely neglect these receptor-ligand complexes. So if I switch notation, just so that we don't have a proliferation of subscript, and I denote by U the concentration of the bulk ligand, and W the concentration of surface receptors, then my model now is basically, it's very similar to the one we just heard in the previous talk. A ligand diffuses in the bulk, it finds a receptor on the surface, they react, and they effectively annihilate each other. We completely neglect the receptor-ligand complexes. So I have this equation now in omega with these boundary conditions for the bulk ligand concentration, and I have a surface equation now which looks like this. And I'm interested in the case when this parameter epsilon is very small. And then for this equation here, we can show that if you give us bounded positive initial data for U and W, then there exists a solution pair in the usual spaces for weak solutions to parabolic equations, so L2H1. This subscript ED denotes functions which take value UD on the outer boundary D0 omega, and the time derivatives are in these spaces. Furthermore, we can show that the solutions remain positive and that they're bounded by their initial data. And the idea of the proof is to cut off this reaction term so that it becomes globally Lipschitz, and then use a Galerkin argument, and you can get all the estimates out. For uniqueness, if we assume that we have classical solutions, then you can use this sort of invariant region argument of smaller to show that your solutions that solutions satisfy these bounds. That this region here is invariant for the solutions of the equation, and then you can use the truncated reaction and you get uniqueness. But there is this assumption of classical solutions so far. So now we're interested in what happens when we make epsilon very small. And so the first thing we did was do some numerical simulation of the equations to try and look at what happens with small values of epsilon. And to simulate these equations, well, I have a PDE posed in a bulk domain omega, and I have a PDE posed on one of the boundary surfaces of omega gamma. And there's a very nice method that's been developed for these types of problems, which is the coupled bulk surface finite element method. There's an analysis of this method in this paper by Charlie Elliott and Tom Ranner, where they look at coupled elliptic equations. And the idea of this method is that you do finite elements in your domain omega, and then you have a triangulation of omega, and the boundary faces of that triangulation induce a surface triangulation of gamma. And you do surface finite elements on this induced surface triangulation of gamma. So to give you an example, on the left here of this plot, I'm going to plot my species U in the bulk and my species W on the surface. And on the right, I'm going to plot the trace of U, so the values of U on gamma and the values of W on gamma. W is black, U is red, I guess. And so what you see is that U is almost 0 where W is positive, and W is 0 where U is positive. And that's what we expect the limiting behavior to be, very similar to what we just saw in the previous talk. And so we can also look at what happens when you change epsilon. The most interesting case was the smallest value of epsilon, which is what you just saw, but it looks like as epsilon is getting very small, the supports of the trace of U and W are becoming disjoint. And indeed, as epsilon goes to 0, we can show the solution pair we've just constructed converges to a weak solution of this limiting problem you see here, which is a free boundary problem where the free boundary is the boundary between the set where U, the trace of U is positive and W is positive. And so, okay, to get a notion of solution to this problem, I have to define the extension of a function. So if I have a function eta, which is in h1 half gamma, I'm going to define its extension to omega in this way here. So it's a harmonic extension. Then I can call a weak solution of this problem any pair U and W in these spaces with the time derivatives in these spaces that satisfy this weak formulation here. And I also have that the product UW is 0 almost everywhere on gamma cross 0 t. And we can show that as we take epsilon to 0, the solutions to the epsilon problem I just showed you converge to solutions of this problem. And the idea behind the proof is to get some uniform estimates independent of epsilon. These first ones, so we get L infinity L2 and L2 H1, are straightforward. They just follow from energy arguments. This one is also quite standard. You effectively testing with mod U, or a smooth function which converges to mod U, and then you get an estimate out like this, which is an L infinity L1 estimate on the time derivatives. But in this constant, we're assuming somehow that the initial data is well prepared. So there's an assumption here that the trace of U0 and W0, their supports are disjoint on gamma. And you can also, this estimate here follows also straightforwardly using the positivity of U epsilon and W epsilon. And so given all these estimates from standard compactness results, we can extract subsequences which converge weakly in L2 H1 and for U and W to a limit pair U and W. And then I can use the fact that this space W11 is compactly embedded into L1 to conclude that U epsilon converges strongly in L1 to U and W epsilon converges strongly to W. Moreover, for every value of epsilon, the pair satisfied this formulation here, so it's clear that the limit satisfies it too. And finally, to check that the limiting pair we've constructed are a weak solution, we also have to check that UW is 0 almost everywhere, and that follows from this estimate when I send epsilon to 0. So we've constructed a weak solution to the limiting problem. But then in applications, one actually encounters that the diffusion in the bulk is much faster than diffusion on the surface. So typically, d omega, which is the diffusivity of the bulk ligands, is sort of 3 orders of magnitude larger than d gamma. And we're also going to assume that the Dirichlet, the outer Dirichlet boundary condition on the species U is of this form here. And then I can just rescale the equations like this, and I end up now with this system of equations for U tilde and W. And I'm going to formally drop these terms, 1 over d omega and d gamma, which are very small by assumption, and that leads me to what I'm going to call the elliptic limiting problem. So now everything looks a bit neater if I switch notation once more and I define V to be minus W. And now our elliptic limiting problem is that U, the bulk ligand concentration now, satisfies the Poisson equation in omega. And now I have effectively dynamic boundary conditions in the sense of a differential inclusion. So the normal derivative of U is minus dTV, where V is an element of beta U, and beta here is this set-valued function. And I also have that U is 1 on the outer boundary. Okay, and I lose the initial data for U, but I now just have initial data for V, and it has to be negative for this problem to make sense.
What happens with Laplace Beltrami?
Yeah, so I dropped the Laplace Beltrami because my assumption is that d gamma is very small. So it's just formally dropping this because the surface diffusivity is very small.
That's the right question in this regard. So the second equation is like the first equation on the surface, pretty much. But then you're dropping the second normal.
Sorry, could you? restate the question
To
maybe?
go back to one slide. So, no.
This one?
Yeah.
Okay.
The second equation,
Yeah.
the second line is pretty much like the first one, but restricted to the surface.
So d gamma is small in the second line. Yeah. I'm still not exactly with you.
Okay. I will ask later. If
Sure,
that's something
okay.
really technical.
Okay. So this is the elliptic limiting problem we get to, and I'm going to denote by a weak solution to this problem a pair uv in these spaces such that for almost all times t in 0t we have this weak formulation satisfied and v is in b to u almost everywhere. And so we can construct, given initial data v0 in L2 that's negative, we can show there exists a unique weak solution to this limiting problem here. And the idea behind the proof, it's just a sketch, is that we consider this regularization of beta, so this function beta epsilon, which is now single valued and monotone, and then now my epsilon problem is now given by this guy here. And then for this epsilon problem via a Galerkin argument, you can show the existence of a weak solution which satisfies these bounds, so L2H1 for u, L2 for beta, and L2H minus a half for the time derivative of beta epsilon, u epsilon. And then from standard compactness results, you get weak convergence in L2H1 of u, weak convergence of beta epsilon, u epsilon to a limit v in L2, and weak convergence of this time derivative of beta epsilon, u epsilon to dtv in this space. And then once again, it's clear that the limiting functions satisfy this weak formulation, as it held for every epsilon. And finally, we have to check this last condition here that, going a long way back, the v is in b to u. And the way we do that is again similar to what we had in the last talk. We have this fact that our choice of regularization yields that in the limit, this limit v has to be negative almost everywhere, and u has to be positive almost everywhere. And then finally, you use the fact that beta epsilon is monotone together with these properties here, and you can conclude that this pair uv, their product is 0 almost everywhere on gamma plus 0t. And so we've constructed a weak solution to the elliptic limiting problem. And so the final thing I wanted to tell you about is how we reformulate this elliptic limiting problem as a variational inequality where time enters as a parameter. And the idea behind this is to define this transformation here. So I define z to be the integral in time of u. And then formally I see that z satisfies these equations here, so just integrating in time. The only claim that one has to check really is that v in b to u implies that v is in b to z. And to show this, essentially we show it by showing that the support of v is non-increasing, and that yields the claim that v is in b to z. And then to get to a variational inequality for z, you know that the z satisfies this weak formulation here for all test functions in the space h1 e0. So then if I define the set of functions in h1 that are positive and take the value t on this outer boundary d0 omega, and for any test function in this set k, I have this being satisfied because eta minus z is in h1 e0. And then I use the fact that from the equation grad z, the normal derivative of z is v0 minus v, and so I end up with this here. Finally, I know that z is positive as u is positive, I know that v is negative, and I know the product zv is 0 because v is in b to z. And so I arrive at this elliptic variational inequality where time enters as a parameter. Okay, and we can also solve the elliptic variational inequality, and we do so with a projected SOR type of algorithm. And so now I'm solving this guy here, so all I'm solving for is z. But of course, I can post-process z, so I can differentiate it numerically in time. So I can differentiate it numerically, and that gives me u. And I can also post-process to get w as well. So because z is not that interesting, in this graph I'm plotting u and w. In this picture I'm plotting u and w. And you can somehow see that it looks similar to the epsilon problem I showed you earlier. So we get the support of w is decreasing, and u is 0, where w is positive. Okay, so that's pretty much it. Just the things we're working on now are the numerical analysis of these numerical schemes, and also dotting the I's and crossing the T's on some of the results I've told you about today. And also, I mean, I told you in the applications, a lot of the cases we're interested in are cases where the surface moves. So if you think of chemotaxis or adhesion, then the surface gamma is time dependent. And I think this can all be extended to the case of a moving surface, which is relevant to applications. And in particular, there's a very nice preprint that's on the archive by Alphonse, Elliott, and Stinner, where they develop an abstract framework for treating PDEs on evolving spaces. And I think using these ideas, we should be able to extend this to the case of a moving surface. Okay, so with that, I'd like to thank you.
For this talk, questions? I was missing some movies, by the way. But
I felt scared after the 3D movies yesterday. I didn't think I could live up to them, so I did so well.
okay. Some more serious issues. So if this is not the case, thanks again, and thanks to all speakers.